So, all right, what we're going to be doing, so we've come a long way. We've had, I think, 5 lectures now on the respiratory system. And so what we're going to be talking about is more systemic things than we've talked about in the past. We've talked about some regulation at the local level, right? How we regulate smooth muscle, right? Using epinephrine, histamine, and these sorts of things at the local level. We're gonna talk about how we make adjustments for the gases fluctuation in the body. Okay, so we'll start with sensory reception. And in the past, when we talked about blood pressure and the cardiovascular system, you remember we talked about those carotid sinuses, right? Remember those things? So there's a little bulge up here, carotid sinus. There's also aortic sinuses. Well, we have a counterpart here, not for pressure, but a counterpart for those carotid and aortic sinuses that are called the carotid and aortic bodies. Okay? And these bodies are the sensory receptors for 3 things, okay? Carbon dioxide, oxygen, and hydrogen ions, okay? Good afternoon. So we're going to be talking about those for a little bit. The cells that are actually present here are called glomus cells. And glomus cells, their claim to fame, every cell has a claim to fame, but glomus cells are the most perfused cell in the body, okay? And like any other claim to fame, it's not entirely true. The endothelial cells lining our blood vessels obviously get more blood and more oxygen, okay? But of all the rest of the cells, they've got a huge, really outsized blood supply, okay? And so what this little body is structurally is a whole bunch of little cells that are just surrounded by capillaries, okay? So those red blobs there are the capillaries surrounding our cells, okay? So in other words, there's a really high ratio of capillaries serving a very small number of cells. Okay? So what do? These are sensitive to the gases in our blood. You know how I like to get into cell biology, right? We are not going to here, but I want to tempt you to take a look and see how this works. Yeah, you know it. Yeah, don't worry about this. But maybe spring break, sitting on the beach. Why not? We know actually quite a bit about oxygen sensing, and just in 2019, a Nobel Prize was awarded for understanding how we sense oxygen in cells, more in a generic sense. But here in the carotid, we've got a very specific purpose for that oxygen sensing, which is the regulation of the whole body. Okay, the whole body's respiration rate. Okay, so we know quite a bit up here. There's actually a little bit of confusion. You'll notice there's a bunch of, I don't know, can you read that? Looks like Louisiana. This right here. We don't actually know, is it all of these mechanisms? Is it some of these? Is it additional mechanisms that aren't listed here? It's not entirely clear. There's been a lot of research on this that took place early last century, and then people just sort of dropped it. But in terms of the cell biology, the cells themselves are sensitive to 2 things. They are sensitive to oxygen, which we'll talk about for a moment here, and they're also sensitive to hydrogen ions. Okay? There's no direct sensor for CO2. We'll talk about that in a moment. Okay, so here's a little chart showing us what's going on with regard to these chemoreceptors, the glomus cells, and their sensitivity to changes in oxygen. Okay, and you'll notice what's on the vertical axis here, y-axis. Minute ventilation. What does that tell us? The rate of what? Sorry? Yeah, breathing, okay? That's a good way to put it. This is the rate of breathing. So how much air are we moving in and out? Okay. So, and we've talked in the past how we calculate that, right? Tidal volume times breaths per minute. So you'll notice that we've got kind of a flat line here. What's our normal arterial PO2 as we're sitting here, most of us?
60.
What's the normal PO2 in our arterial blood? You remember? Yeah, 100. Okay. So normally we're sitting right about there. Okay. And if oxygen levels go up or they go down a little bit, we really don't make any adjustment to our breathing rate, do we? And it's really only around 60 millimeters of mercury that we start to make a substantial change. Okay? So we can say that our carotid bodies are relatively insensitive to changes in oxygen. Okay? And specifically have normal levels and pretty wide variations of normal. It's not until we get down to about 60, which is pretty low. So how low is this? Right? It's pretty low. You remember our hemoglobin, oxygen hemoglobin dissociation curve? Okay. Where does 60 put us? How much oxygen saturation do we have at 60? Yeah, and this might be why, right? This might be why it's really not that important to micromanage our oxygen levels. And it's because we still have about 80% saturation of hemoglobin, okay? And yeah, we've got plasma levels down, 40%, or I'm sorry, down 60% if we're at 60 millimeters of mercury. But we've got a ton of oxygen left on the hemoglobin, right? Okay, just for perspective, right? How many of you have been to Mount Mitchell? Yeah, not far from Asheville. It's a hike, right? The atmospheric pressure there is about 600 millimeters of mercury, right, compared to sea level, which is 760. So how would that change the oxygen atmosphere? What would be our PO2 in the atmosphere? Okay, can somebody tell me how you calculate that? Multiply what by 0.2? Okay, so we're going to she said we're going to multiply the atmospheric pressure times the percentage of oxygen in the environment, in the atmosphere. Okay, so we'll say oxygen is 0.21. Okay. All right. So I'm not testing your ability to calculate, but I do want you to understand how the calculation works out. And we've talked about partial pressures, right? So this comes out to be about 126. Okay. And I'm going to estimate. All right. So remember this PO2 here in the atmosphere. It's one of the determinants of the alveolar PO2, right? And then PO2 of the alveoli is what we really care about, right? Why do we care so much about the alveolar PO2 more so than the atmosphere? We exchange gases at the atmosphere or at the alveoli? Alveoli, right. Okay, so we're exchanging gases across the wall of the alveolus. Okay. Okay, so I'm going to estimate this, just sort of the back of the napkin sort of thing. It's about 65%. Okay? This assumes we're not making any compensations. All right? Okay, so here's the big question. The rest was just calculation. Okay? Here's an important conceptual question. If we've got a PO2 in the alveoli at 8, what is the PO2 of the arterial blood that's leaving the lungs? This goes back a couple lectures. 80, good. Okay, whatever we have in the alveoli, we're going to reach equilibrium. Okay, and we talked about why that happens. We're always going to reach equilibrium, okay, because of how efficient the lungs are with their massive surface area, right? Okay, 8. So even if you go all the way to the very peak of Mount Mitchell, are you going to have a response? You won't. Okay? You're going to have to go west, right? You go to Denver, right? You could go to someplace at a much higher altitude, okay? 6,000 some feet is not nearly enough to have any kind of response, okay? Are we gonna carry less oxygen in our blood? Yeah, a little bit. But we've got a lot of hemoglobin there, right? Here's another test, right? Hold your breath. Okay. I couldn't find a good chart that went beyond 30 seconds. I don't know what's wrong with these people. We could do this in the lab. But if you extrapolate here, you hold your breath about a minute, you're probably down to the range where you're gonna have a response, okay? So holding your breath for a minute sure feels like you've got a problem, right? Should we try it? No, okay. Right. But that's not due to oxygen so much as it is carbon dioxide. Okay, now, carbon dioxide is something we really do micromanage. Okay. And as I said before, it's not CO2 we're detecting directly, it's the hydrogen, okay? We spent way too much time talking about this equation in gas transport last time, right? But every time we add CO2, we're gonna get hydrogen, right? Every time we take away CO2, we're gonna lose hydrogen, right? So hydrogen's a great proxy for CO2 levels because of the efficiency of this equation to reach equilibrium. Okay? But how much do we have to change PCO2? They're really small differences, right? Anybody have a reason maybe why this is important? Why do we care so much about CO2 and not so much about oxygen? CO2 is a waste product, right? We don't need CO2. Why do we care about its value so much? Yeah, got an idea?
I mean, it does form an acid, which would affect the overall pH and maybe the folding of enzymes.
Yeah, that's a great explanation. I can't do better. It's going to affect pH, okay? And pH affects, as you suggested, the folding of proteins to form enzymes and other structures. So much of the body's protein structure relies on a certain pH, okay? And not only proteins, okay? So we've got these chemoreceptors that respond to CO2. CO2 goes up or goes down. What else are they going to be able to detect that doesn't have necessarily to do with CO2 itself? Is there another source of this H plus that could occur in the body? Yeah, right. So what are some things that make acid in the body? Or make your body acidic? Alcohol. Not at your age. What else? Yeah. Lactic acid's kind of a big one. Yeah, when it comes to exercise, we'll see that at least with strenuous exercise, you can generate lactic acid. Okay. And there are other things like diarrhea that will get rid of a lot of bicarbonate that way and alter the pH of our bodies. So there's lots of things that can cause hydrogen levels to change independently of a CO2 cause, okay? So we can respond to acidosis and alkalosis in the body. Okay, so I've got a question for you. Pull everywhere behaves. So given what we've talked about, which of these scenarios here would provide the strongest stimulation for a change in our rate of ventilation? Okay. So take a moment and read through this. And then talk to somebody close with you. We'll give you a couple, 2 minutes or so to talk through this and see if you can come up with the correct answer. increase in venous PCO2. That might mean we have an increase in arterial PCO2, but we don't know that, right? We might have efficient gas exchange and we get rid of any excess. Okay, so all our body knows is what's in the arterial blood, not what's in the venous. There's no chemoreceptors for the venous circulation. Yeah.
Is it like a whole, is it a closed system? So wouldn't it like violence all be like this?
You mean to say that wouldn't the arterial and the venous PCO2 be the same?
Yeah, just because it's like all the system.
So because the venous blood is going to pass through the pulmonary capillaries, we'll get some gas exchange, and that's where the PCO2 is going to change. Okay. So normally, we talked about a value of 46, remember that? Where the CO2 comes down to about 40. Okay. So it goes from a high level to a low level at the pulmonary capillaries. So we are going to get some change between venous and arterial. Yeah. Yeah, so the 5% change in arterial PCO2 would certainly matter. 5% is, by the way, that's about 2 millimeters of mercury, right? Difference. And we can certainly see that there's going to be a ventilation change there. Okay. We do have one additional location for chemoreceptors. Okay, so we talked about arterial arterial chemoreceptors in carotid bodies. But we also have, in addition to those peripheral chemoreceptors, we also have central chemoreceptors. These are found within the brain itself. So sort of scattered throughout the medulla, we've got various cells that seem to be sensitive to, again, the hydrogen ion concentration. Okay? So it's not CO2 directly. But we've got an interesting situation here that makes these a little bit different. Okay? So remember the blood-brain barrier from maybe 252, biology 252? What do you know about the blood-brain barrier? Barrier, right? Yeah, it's very selective. Astrocytes are surrounding those endothelial cells to stimulate tight junction production and make sure that we've got really, really low permeability to ions and other compounds passing through the capillaries of the brain. Okay, so one of the things that can get through is CO2, right? CO2 doesn't need to pass between cells. It's a gas. It goes right through them, okay, as we've talked about before. But what happens to hydrogen ions? As you can see there, they don't get through. Okay, so the only hydrogen ions here that are going to make it are those that are produced from CO2. Okay, I'm exaggerating a little bit, but by and large, we have CO2 being able to make it through, and so CO2, once it goes through the blood-brain barrier, will be converted to bicarbonate and H+, right? And we get hydrogen ions that can then stimulate central chemoreceptors. Okay. What about metabolic acids, lactic acid, and these sorts of things? Yeah, they do not easily pass through the blood-brain barrier. Okay, so if we've got lactic acid generating lactate, hydrogen ion, right? That hydrogen ion is not going to pass to the brain. Okay, so this is the distinction. So the chemoreceptors here in the central chemoreceptors are generally considered to respond only to CO2 changes. And the peripheral chemoreceptors respond to both, right? Metabolically generated acids as well as CO2 generated H+. Okay, so the next section we're going to talk about is how do we regulate this process, right? We have some sensory receptors. What do we do with them? How does this whole process work out? Well, your book goes into a bit more detail here than I think is probably necessary. Usually, as you know, I go into more detail in a lot of areas. But This is an area of science which is really evolving. And if you look at a textbook from 2 years ago and from 5 years ago, 10 years ago, you'll see a different story each time. What we understand generally is that we have some respiratory centers in the medulla, much like we have cardiovascular centers in the medulla. Remember those? Okay. So just like cardiovascular centers, they're going to respond to a bunch of things and then have some sort of output. Okay. So the output here from these two sets of nuclei in the brainstem are in charge of sending stimulatory signals to the respiratory muscles. Okay? So we broadly define or broadly categorize these as inspiratory muscles, right, and expiratory muscles. Okay. All right. So the two regions here are the dorsal respiratory group and the ventral respiratory group. The dorsal respiratory group is responsible for sending signals to inspiratory neurons, and those are neurons that will ultimately lead to inspiratory muscles. And the ventral respiratory group, or the VRG, sends signals to the expiratory muscles. Okay. Probably the most important thing to note here is that the VRG is responsible for the intrinsic rhythm. Okay, so we've got some cells here that function very much like the pacemaker cells we've learned about with the heart. Okay? So there are cells with pacemaker potentials, right? Remember that whole process? They don't use the same HCN channels and all that stuff we learned about, but they function similarly in that there's a rhythmic production of action potentials. And these two regions interact with one another, but it's the ventral respiratory group that is the one setting the rhythm, and the dorsal respiratory group will often be the one to modify that rhythm. Okay. So, when we look at a few of the nervous structures, we've got the phrenic nerve at the top. What is the phrenic nerve innervate? The diaphragm, right. External intercostal nerve, external intercostal muscles, right? So these are the muscles that tend to be active during breathing, okay? And look at the top here, it says quiet breathing. So we've got some relaxed breathing going on. And there's a small involvement of the external intercostals, but it's mostly the diaphragm that's being stimulated, okay? So which of our respiratory centers is sending those signals? If both of these are involved in inspiration. Yeah, the dorsal respiratory group is sending signals down. Okay, rhythm's being set by the VRG, but the DRG sends the signals on down. Okay, and we have production of tension, right, periodically, and then it relaxes. Okay, and the lung volume goes up, goes back down. Right. All right. Here's the diaphragm. External intercostal muscles. Right. The internal intercostal muscles are a little bit more often involved in expiration, okay, but not relaxed expiration. Relaxed expiration is entirely passive. But if we need to force air out of our lungs, next time we're shouting at the ball game, right? That's our internal intercostal muscles helping us out to provide some compression to the thorax. Okay. But all these signals are going to be originating from the DRG and VRG in the brainstem. Okay. So every time we breathe in and breathe out, we're sending signals to the muscles to either contract, or different sets of muscles to contract at different times. Okay. Now, that's all pretty straightforward. What sort of inputs can modify this? We've got a bunch here. We're not going to spend our time focusing on all of these. But you can imagine, we have voluntary control, right, over breathing. We could breathe in anytime you ask, right? I can take a breath. I don't have to wait for a particular part of it. I can do it anytime, okay? No problem. Coughing, sneezing. There's a stretch reflex if your lungs get inflated too large. every time we speak, right? All these things are overriding the activities of the DRG and VRG. Okay? And those come through some nuclei in the pons. Okay? Importantly, we've got chemoreceptors. Okay, so our chemoreceptor input is one of many, okay, but it's probably the most important of these, right? We could probably live if we didn't have speech. We could live without some of these things, right? But day to day and hour by hour, we're making adjustments because of the chemoreceptor input from the central chemoreceptors as well as peripheral. Okay, so real quickly, the whole reflex. We've got chemoreceptor changes in gases, or H+, right? Sensory input comes in. What are the cranial nerves, by the way? What's carrying these signals? I'm giving you a hint. Yeah, glossopharyngeal nerve, and what's the other one? Vagus. Okay. So why do those look familiar? Same as carotid sinuses, right? Okay. All right, so we've got some sensory input, and we've got a motor output to those muscles of diaphragm, intercostals, right? And where does this all end? Well, it ends with changes in the rate and depth of respiration. Okay. Why do the rate and depth of respiration matter? What changes when we alter the rate and depth of our respiration? What changes in our lungs? How much oxygen is going to be in our alveoli as well as how much CO2, right? So, all right. We've got a little activity here we can run through. So, initially, let's just talk about the immediate changes that happen when you go to a high altitude, okay? So let's not mess around with Mount Mitchell, right? We're going to Denali, right, in Alaska, and P, pressure of the atmosphere, is only 349, okay? So let's say you haven't made any changes yet, right? We're gonna follow that change, but what's going to happen as an immediate result of this? So I'd like you to figure out what the PO2 is going to be in the atmosphere. You can estimate using that 65% rule that the alveoli are going to be about 65% the PO2 of atmosphere. Okay. And tell me what these PO2 values are going to be throughout the system. Okay. And then we'll address that last question together. Okay, so take a minute or two, 2, 3, see if you come up.
No, that's oxygen.
All right, so let's take the easy part. We can calculate the PO2 in the atmosphere, right? Mm-hmm. Right. We know what a partial pressure is. What is the PO2 in the atmosphere? What'd you come up with?
73. Right?
Give me a number. 73. Did I come up with that? Oh, I gotta write it down. Oh my gosh, hang on. 73, we'll go with that. Does everybody agree? Okay, 73 millimeters of mercury. Okay? And let's say we're ventilating normally like we do when we're at sea level. Okay? And we can estimate about 65%. So here's where we estimate. What do we get for the alveoli? 48 millimeters of mercury. Okay. So that's uncompensated, right? We haven't made any adjustments. We're breathing as we normally do. We breathe in some fresh air that mixes with the quote-unquote stale air that's already in our alveoli, right? Remember the previous 2 lectures ago? Okay. So now, this is a key thing to understand, right? What happens next? What's going to be our PO2 in arterial blood? That is the pulmonary veins, blood that moves on to the arteries of the body. What is this value here? 48, okay? Always gonna be the same as what's in the alveoli. All right, unless we have some pathology that interferes with gas exchange, okay? But we're gonna have a PO2 of 48, okay? So what's the carotid artery gonna see? The carotid bodies. What's gonna be the value there? Yeah, I heard a couple of you asking about it. It's gonna be 48 as well, okay? So whatever the alveoli have as their PO2, we can make an assumption that what's going to happen, at least under normal conditions, is that arterial blood is going to come up to the same value, and then the carotid artery contains arterial blood, so it's going to come up to the same value. Yeah.
Would it not be like 5 less because of the ventilation perfusion?
Okay, so great point. She says there's going to be a slight ventilation perfusion inequality. We talked about that. So there's a little bit of drop here, but assuming that, yeah, we're gonna sort of sweep that under the rug for now. Okay, I'm glad you know that, and we talked about that a little bit, but there's also a little bit of venous mixing that takes place from bronchial circulation, which is different from pulmonary circulation. Ask me about it some of your office hours, I'll tell you all about it. But for rough estimate purposes, we are going to have pretty much the same amount of oxygen here, okay? So, oh, yeah.
In the full everywhere though, you said that because the venous blood goes and then becomes oxygenated, it wouldn't be the same as it wouldn't be like this, it wouldn't have the same piece, as it is in the artery, so why is it the same PO2 here between the venous blood and the carotid artery?
Okay, so the same thing happens as with the example with carbon dioxide. So carbon dioxide, if carbon dioxide is elevated over here on the left, it's going to reach normal levels here. Normal being defined by what's in the alveoli, okay? And the same is true for oxygen, so this is really no different. We have some changes that have happened in our blood, right? So, just so we're on the same page here. This PO2 30, this reflects metabolic activity of cells, right? So metabolic activity of cells consumes oxygen and lowers it. So when that blood comes up to the lungs, it's going to be lower, right? And when we pass through the lungs, what happens is we equilibrate PO2 values with alveolar air, okay? And the same is true of CO2.